Marco, take it away.
Thank you, Nancy. And a very good morning to you all. I hope you had fruitful discussions in the small groups related to our case study of a clinical trial protocol. Our intention this morning is to actually follow on what we started yesterday. And I would like you to keep in mind the ladder of the levels of evidence. And one of the take-home messages that I wanted to emphasize yesterday was you always try to reach for the highest possible realistic level of evidence when you develop new treatments. We're going to be discussing how to conduct a clinical trial, or maybe rather we will discuss what kinds of typical designs there are for clinical trials. What are their weaknesses and what are their strengths? You have there what we call the gold standard. That is the randomized controlled trial. And we will go through each of those words individually. Randomization, what does that mean? It means that the study participants are identified based on clear, predefined criteria. And this is done in a study phase called the screening. It's called the screening procedure. So you want to make sure that the participants entering the trial will have the desired characteristics. Then, once you have participants selected, they are divided, the participants are divided or allocated to treatment groups randomly. Again, the typical regulatory scientific jargon that you will see all of the time in protocols, and it's important to understand what the words mean. And in the process of randomization, we typically today have computerized systems. So the computer will give you a code that is given to that specific study participant, and the computer takes care of the randomization process. In the past, when we did not have computers, what did we do? We had closed envelopes prepared by someone who was not involved in the clinical trial conduct. That was one way of doing randomization. Now, there are a lot of different procedures that can be used when you do the actual randomization, and I'm not going to go into that level of finesse, but it's important to understand what the process is about. So at the end of the day, the important outcome is that neither the participant nor the investigator can influence the treatment allocation. So that is one way of reducing the risk of what? Bias. Very good. Other advantages of randomization, well, you avoid the selection bias we already touched upon. The possibility that the treatment outcome is influenced by the selection. It could happen that, for example, if you have an open label study, you know what you're receiving. The investigator may bias the allocation by perhaps allocating certain types of patients into placebo group. And that will cause, obviously, bias to the study results. The process also makes sure that the treatment groups within a clinical trial are balanced. In other words, they are similar and that only the treatment is different in the group. So that is really the core of the matter. Only the treatment is supposed to be different. If you have group A receiving new investigational treatment, group B receiving placebo, you want to make sure through randomization that only the treatment is different. The population in the two groups is the same or has similar characteristics. Now, sometimes it may be important to take a further step in the randomization process. Now, let's think about a lung disease. And let's think about the reality of there being smokers and non-smokers. Now, for a lung disease treatment, whether or not you're a smoker, it might have a big impact on how the treatment works. So you want to make sure at the beginning of the trial that in the two treatment groups, you will have the same proportion of smokers in both of the groups. And the way to do that is to stratify the randomization. We call it stratifying the randomization based on your smoking status. So we collect that information in advance and we take that into account in the randomization process, ending up in having maybe 10% of smokers in both treatment groups, and all is well. Smoking will not introduce any bias into the results. Then what we oftentimes see is that there are still, even if we do randomization, there will still easily be some differences in the groups, like there could be a high number of males in one group or higher mean age of patients in one group. But if you've done your randomization well, those differences at the end of the day are easier to account for in data analysis if there are only a few. But if you don't do randomization, or your randomization fails, you will have a number of differences, and then you start to have a problem of having many imbalances in the study population between, in this case, the two groups. Are there any disadvantages? Well, it doesn't guarantee that the study is free from bias, but it's an effective means of reducing the risk of systematic error. And randomization is no guarantee, particularly if you have an open label study. You know what treatment you're receiving. You may have an effect on the compliance to the treatment, receiving other treatments, dropping out of the study. Who wants to be on placebo? Who wants to be on treatment that is perceived to be less efficacious? You will easily have an impact. If you have an open label study, you know what you're receiving. That can impact the undesirable effects that you report. Now, it's important to make a distinction between adverse events and adverse drug reactions. An adverse event is any untoward event in the course of a clinical trial. If you walk out the door and you're hit by a car, that's an adverse event. If you walk out the door and you're hit by a car, but you were taking a pill that made you drowsy before you went out, now that is an adverse event, but it also can be an adverse drug reaction. You weren't paying attention because you were drowsy. So that is an important distinction. And if there's a possibility that an adverse event is caused by the treatment, it's reported as an adverse reaction. So it's important when you review the results of clinical trials, it's important to pay attention to the terminology. Adverse event is any untoward occurrence without assessment of causality. Adverse reaction is an event where at least there's a possibility that it was caused by the treatment. So open label studies we want to avoid, but sometimes they are unavoidable. And there are ways to reduce the risk of bias even in those situations. I will return to these in a moment. But we want to do the studies under double-blind conditions whenever possible. There are non-standard situations. And when we talk about non-standard situations, again, thinking of the ladder of evidence, we want to do studies in non-standard situations, again, up to, corresponding to the highest level of evidence that is possible. Conventional development means that you do your phase 1, your phase 2, your phase 3, then you seek marketing authorization, you get the license, and then you do your post-authorization studies. So that's the conventional way forward. There are unconventional developments when reality hits you hard and tells you that it is not possible to do a big randomized clinical trial. Then you may have to do an open label study. One typical situation where an open label study is done is in advanced cancer. In a situation where you don't have any identified treatment options left, you do an open label study. And that may suffice for marketing authorization. I referred to yesterday to the extreme situation where it's impossible to do clinical trials, the anthrax case, where all you will have is animal studies showing that the treatment in all likelihood will be effective and will prevent death in human beings if they're exposed to inhalational anthrax. But it's important not to become too distracted by these examples. What we always think is to aim for the highest level of evidence that is possible in the clinical situation. Medicines for use in children. It is clear that you will want to reduce exposure to placebo. Whenever possible, use a control treatment that is an established one and compare the new treatment to that. Or, as in the stiripentol case, all of the patients entering the trial were on effective medication. What you did was you added either placebo or stiripentol, the new drug, on top of the combination of effective treatment. So in that situation, placebo is not an ethical or a medical issue. It's important to, and it's recommended reading to look at the CHMP guideline on clinical trials in small populations. So if you haven't done that already, that's a recommended bedtime reading. You will fall asleep. I can guarantee, I did. There's also another note for guidance, and that's on clinical investigation of medicinal products for the pediatric population that dates back to 1999. It still makes good reading. And there are updates, and there will be updates to the therapy area-specific guidelines. There will be pediatric addendums to guidelines on cardiovascular disease, diabetes, and so forth, and schizophrenia, which is also a disease that is diagnosed in adolescence. So we moved to we're past double-blind, randomization. We're going to the controlled word. The control group, obviously, is your yardstick against which efficacy and safety can be evaluated. And, of course, it means a lot how you choose your comparator. Particularly the US FDA is very fond of placebo control. Placebo control gives you perhaps a cleaner picture of what the new treatment does in terms of efficacy and safety, but what it doesn't tell you is how the new treatment fits in with what we already have. How does it perform in comparison to what we have out there? There are ways that you can get an idea how the new treatment works in relation to available therapies without direct comparison. Again, we would then revert back to what we discussed yesterday about meta-analysis. So if you have treatments A, B, and C available, and those have been approved based on placebo-controlled trials, and you have new treatment Z that will be compared against placebo, but not against the available active treatments. You can do a meta-analysis and get an idea of, based on the drug placebo difference, and where this new treatment falls compared to what we have. But it comes with the caveats of meta-analysis. It comes with the caveats that you don't have direct comparative data. Placebo, if ethical, if medically acceptable, is still used a lot, but there are more and more demands against using placebo. Particularly in Europe, there are many centers that will simply refuse placebo-controlled trials in many therapeutic areas, such as those related to psychiatric conditions. They will only accept active control studies. Whereas the US FDA may have significant issues with accepting active control studies. And they're mainly worried about whether it's possible to reliably demonstrate that the new treatment, in comparison to the available treatment, is not inferior. It goes into statistical discussions and possibilities to rely on a non-inferiority outcome. But then in Europe we have tried to be active and promote other types of studies. And we very much like a situation where you have an active comparator, an established treatment, you have the new treatment, and you have placebo. So you have both placebo control, if feasible, and you have an active comparator in the same trial. And we think that the problems are washed away, the problems that the FDA has. Continuing to talk about control, well, standard care plus placebo is exactly the add-on situation that we had with stiripentol. Then what do we mean by best supportive care? If you read through studies done in cancer, particularly the advanced cancer where no available treatment can be identified, the comparator is often best supportive care. Best supportive care is mental health. It's providing best possible relief for the symptoms, pain, nausea. It's dealing with nutritional issues. It's dealing with the well-being of the study participant as far as possible without giving any cytotoxic treatment. We still do those trials, and if it's possible to conduct such a trial, it gives probably more reliable answers than doing a trial without such a control arm. But for obvious reasons, there tends to be a high dropout rate in those trials because there are always more possible treatment options coming under study, and patients from the best supportive care group will drop out and they will want to try the other investigational treatment. So that is an issue for the interpretation of the trial, that patients tend to drop out. And then the established therapy. When we talk about established therapy, we usually mean an authorized treatment, something that's on the market, but occasionally, and particularly in cancer, you may have what we call evidence-based therapies that have never gotten a license, but they're widely used. They're widely in clinical use and therefore considered an acceptable comparator. It may just be that the cancer treatment in question is very old. Its patent has expired. The company who has the marketing authorization has never wanted to send in the money to get the additional indication, but it's still widely used. And if it's known to be effective based on clinical studies, it can be accepted for regulatory purposes as well. The next word, blinding. It decreases the risk of bias resulting from knowledge. It may be a single blind situation. In other words, maybe the patient knows what he or she is receiving, but it's important that when the investigator is doing the assessment, the investigator doesn't know. But of course that is a situation where it's ever so easy to break the blind, intentionally or unintentionally. So it's not a very good safeguard, but sometimes, it's better than nothing if you can't really blind the treatment double dummy. Double dummy is a setting where, if you assume that your control treatment is a tablet and the new treatment is a capsule, well, you can tell the difference between a capsule and a tablet. So what do you need is, if you're comparing to placebo, you need a placebo capsule and you need a placebo tablet. So it's possible that each patient will receive a tablet and a capsule, but with different contents in them. So that is what we mean by double dummy. So you can't tell based on the appearance of the of the treatment. If particularly it's important if we cannot blind the study, if we have to do an open label study, such as in certain cancer treatment settings, you will want to have a blinded central committee looking at the objective results. In cancer, you will have the X-ray, you will have the CT scans, you will have the MRI, you will have the PET scans. Those are sent to the adjudication committee or the evaluation committee. They will judge whether the patient is having a treatment response or not, without knowledge of the treatment group. So this is a good way to evaluate objective data without having to blind the investigator and the patient. Sure.
What kind of people are usually in such a committee? Are they not involved in the trial, and how are they selected?
Right.
Is it on? I was just wondering who's selected to be in such a committee, and are they involved in the trial, or are they completely independent?
They are completely independent. They're not allowed to be involved in the conduct of the trial in any way. So they are independent, they are remote, and they are firewalled. You may also have this sort of committee in large clinical trials that are completely double-blind, but you still want to have an independent board looking at clinical outcomes such as myocardial infarction, heart attacks, stroke, heart failure, and so forth. You want to have an independent, objective evaluation without the subjective elements that are easily brought in by investigator evaluation. Typically, large studies looking at the effect of treatments for diabetes on cardiovascular outcomes. Is the risk of stroke, heart attack increased, decreased, or is there no effect? Then typically you will have an independent data monitoring committee, IDMC, or data safety monitoring board, alternatively called, who will have access to unblinded data mainly for the purpose of safety monitoring, but also for interim analysis. You may have interim looks at the data during the course of the study on the proviso that those interim analyses are planned in advance, so that they're stated in the protocol that at point X you will have an interim analysis. And it explains how the interim analysis is done. It explains how the results of the interim analysis are safeguarded so that they do not reach the investigators or any other people, but those who are eligible to be involved in the process of interim analysis. An interim analysis may lead to the data monitoring committee to recommend study closure. In other words, that the study is stopped. And there are also those situations, unless they are related to safety, They're supposed to be explained in the protocol in advance. So the study may be stopped for overwhelming efficacy. And if you're showing in an interim analysis an overwhelming degree of efficacy, it's probably unethical to continue the trial and continue to give maybe placebo to half of the patients. The study should be stopped and all of the participants should be offered the possibility to get the effective treatment. Or alternatively, there may be an issue of not seeing any hint of any treatment effect at all. And in that situation, if the interim analysis is done at a good point in time, it would probably be unethical to continue the trial as well, because if you're never ever going to show an effect, if the trial run for 50 years, it's futile to continue that. So that is a futility decision. And then, of course, you may have a safety issue which either leads to temporary closure of the trial or permanent closure of the trial. We move on to planning and designing clinical trials, and this is really the simplest possible way of depicting what a parallel groups clinical trial scheme looks like. So you will have the screening period, as we discussed. You look at look at the eligibility criteria. Then you randomize the study subjects that are eligible for randomization. You begin treatment. You have different treatments with lighter and darker blue, and then you have a red treatment. And then you have your your study endpoint, after which the treatment may be stopped, or there may be a follow-up after treatment to find out if there are any safety issues during the follow-up, how quickly the efficacy is lost, and so forth. So this is the most typical clinical trial scheme that you will be familiar with. But then we have another classic example, and that is the crossover trial, where you have, in this case, a simple 2x2 crossover, where you have two treatment sequences, A and B, and you have two treatment periods, 1 and 2. You screen the subjects, you randomize them either to sequence A or to sequence B. In sequence A, all subjects will first receive the blue treatment, then they will cross over and end up having the red treatment. Whereas in sequence B, they will first receive the red treatment and then cross over to the blue treatment. So all the participants will receive both study treatments. And the participants will actually serve as their own controls. So you know what happens in an individual who is receiving the blue treatment and the red treatment. But that's not where it ends. There is a possibility that you will have a different treatment effect in period 1 and in period 2. How do you explain that? How do you explain the possibility that the blue treatment is working differently in period 2 than it is in period 1?
Maybe the treatment you're giving in period 1, so the red treatment affects whatever's happening when you're giving the blue treatment, or vice versa.
If we assume that the blue treatment is the active one, it may carry over. The treatment effect may carry over to period 2. We call it a carryover effect, and that is why in these types of trials you have a washout period between period 1 and period 2. And the duration of the washout period depends on how quickly the drug is eliminated from the body. Well, it is also possible that Well, actually, if you have different effects in sequence A and sequence B. No, sorry, I'm mixing myself. If you have different effects in period 1 and period 2, and you have included a washout period in between, we may end up in a situation where you can only reliably look at the results of the individual periods. Now, what are the circumstances where you can use this sort of a trial? It has the advantage of not requiring as many patients as the parallel groups clinical study. Typically, you would use that in a situation where you have a chronic condition that is not fluctuating a lot from day to day, from week to week, and where you can observe the treatment effect quickly. Situation that could be a situation where typically this sort of study is used is in chronic obstructive pulmonary disease, COPD, where you have chronic symptoms. You do have the exacerbations, you do have the worsening symptoms, but it's pretty chronic and pretty stable from one day to the other. And you introduce a bronchodilating treatment that dilates the airways. You probably will observe the maximum effects within a week or two. And then it is possible in that situation to do a crossover study. You can compare it with placebo, you can compare it with another active treatment.
Chronic stable diseases, if the medication is for cure, then this doesn't apply, because in the recent hepatitis C trials, then let's say the blue treatment is the treatment, then the placebo will have no effect because the patients would be effectively cured.
Certainly for curative treatments this is not an option, but for chronic stable diseases it is one designed to be kept in mind. I'm coming back to adaptive studies for a brief moment later on, but let's look at the study protocol, the study cookbook. What you saw this morning was a one-page summary, synopsis. The protocol itself will typically be around from 80 pages to 120 pages, sometimes more, depending how complex the trial is. So a lot of things go into a protocol, and you will easily spend several days if you want to find out everything about that study. You will first of all see a page that contains protocol amendments, if there have been any. The protocol amendments are changes in the procedures of the study that are introduced after it's initiated or after it's written. And you're supposed to be telling up front to the investigators, to the ethics committees, to the study participants what changes have been introduced. It's important to know, because the amendment may entail that you are changing the eligibility criteria for the trial. The amendment may mean that you have a new safety monitoring system in place because of emerging safety signals in the trial, and so forth. So the amendments are important, and you always have to make sure that you are looking at the most recent version of the protocol. Protocol adherence and protocol violation are important terms. Protocol adherence, obviously, that you adhere to all of the advice that's given to you in the protocol. Protocol violation, they can come in many shapes and forms, and in the worst case they will mean that the study participant no longer can continue in the trial because the violation is such that his or her safety is affected. It could be related to taking the wrong medication, taking medications that you are not allowed to take while you're in the study, and so forth. Protocol describes the study objectives, the primary ones and the secondary ones, and it's important to realize that we judge clinical trials on the basis of whether or not they reach their primary objective, their primary endpoint. So in regulatory jargon, we say that, at least, that a trial is a failed trial if it hasn't met its primary objective. It doesn't mean that it's a worthless trial. Negative trials, failed trials, are valuable ones to take into consideration in the whole context of available information. But it means that it will probably not be very useful for any regulatory, positive regulatory decision. The secondary objectives are supposed to cover aspects that are not covered by the primary objective. Typically, if efficacy is the primary objective, safety will be among the secondary objectives. Looking at the pharmacokinetics of the new treatment can be a secondary objective, and so forth. Or it can be quality of life assessment, other patient-reported outcomes, and so forth. Secondary objectives, important, they're there to support the primary outcome of that trial. All of the study design aspects, whether it's randomized, double-blind, placebo-controlled, active-controlled, all of those aspects are described and also motivated. So the protocol will give you an overview of the disease, it will give you an overview of the treatments available, and it will give you the arguments why this particular design and this particular comparator was chosen. The number of subjects needed for the trial in order for it to reach its desired power, and the inclusion and exclusion criteria. I have to speed up a little bit, I notice that I'm running a bit late. So the outcome measures and endpoints. Again, we talked about the primary and secondary objectives of that trial. We have corresponding outcome measures, primary and secondary ones. The primary measure will be decisive for the interpretation of the trial, if it's a success or not. The secondary measures are intended to provide more information and support the primary measure. They can, let's take Alzheimer's disease as an example. In Alzheimer's disease, if you have mild to moderate Alzheimer, regulators expect that you will show efficacy using two primary endpoints, one being cognition and the other being function. So how you function and what are your cognitive capabilities. Both have to be met for the study and the treatment to be considered a success, but you will have supportive efficacy measures, including quality of life. You will have healthcare use. You will have global evaluation of the patient's status, and so forth. And those are supposed to be supportive, showing supportive effects that support the outcome on the primary endpoints. You will have in the protocol information on how safety is being assessed, if there are any special measures necessary for the evaluation of safety, like if you expect that there will be neurological problems, or there might be cardiovascular problems. There will be a separate detailed description of how those will be looked upon and evaluated. Statistical methods will be described briefly. There will be a separate statistical analysis plan attached to the protocol, usually at a later stage. Importantly, you will have the case report form. We will discuss the contents of the case report form separately and also touch upon those in one of the small groups. Then there will be a description of how the study will be monitored, and that's important also to find out. And we discussed in our small group this morning that particularly if the studies are run globally, if the studies are run in Australia, in Europe, in the United States, in India, in China, in Korea, in Japan, in Taiwan, you probably want to know how the study will be monitored. How will it be made sure that the protocol is being followed, and that there's adequate that the patients are treated in the way that the protocol tells. Protection of patients. Also, in addition to informed consent, the Institutional Review Board and Ethics Committee approval, data handling and record keeping, and then financing and insurance for the participants. When we talk about outcomes, you will often see that we talk about variables. We talk about variables because the outcomes can have different values. They're variable. They can be objective or they can be subjective. Now, this doesn't entail any intention to say that a subjective endpoint is not a good endpoint. On the contrary, they are used usually as mutually supportive endpoints. So an objective endpoint is impartial. It's based on observation. It can be a physical, chemical, or a laboratory measure. And there's low degree of interpretation what it means. It can be your blood pressure. It can be your blood cholesterol level. It can be your C-reactive protein if you're studying an infective disease. Whereas a subjective endpoint is taken as an important measure of perceived benefit and perceived harm. It tells how the investigator feels about the effect of treatment. It tells how the subject, the study participant, feels about the effect of treatment. We talk about hard endpoints. Well, they're hard in many ways. They're hard to show to begin with in clinical trials because they are events that aren't occurring that often. They can be stroke for a treatment of hypertension, high blood pressure. It can be reaching the status of needing dialysis treatment if you have kidney disease. So beginning dialysis treatment is considered a hard endpoint in clinical studies, or death. Or they can be soft or surrogate. And typical accepted surrogate endpoints are blood pressure and laboratory measures of kidney function. And they are surrogate because they are known to predict hard clinical outcomes. If your blood creatinine, which is a measure of kidney function, doubles, it correlates with your risk of developing kidney failure to the extent of needing dialysis treatment or kidney transplant. A certain it requires that a certain magnitude of change is known to be important. Then we have biological markers, or in short, biomarkers. They tell us about the presence and activity of disease or condition. They tell us about the presence of a phenomenon, but we don't know if they have surrogate value. That's the difference. We don't know if the presence of this biomarker or the level of this biomarker actually does correlate with the risk of reaching a hard outcome. You are expected to collect data in clinical studies, in epidemiological studies, to find out if we can say that it's possible to qualify a biomarker as a surrogate marker. The subjective endpoints are typically patient or investigator-reported outcomes of what is considered to be meaningful to the individual. What is a sign of there being a treatment benefit? So it can be pain, depression symptoms, function, activities of daily living, quality of life. And when you measure these, it's important that you check that the study is using recognized rating scales that have been validated for the clinical situation. Otherwise, there will be difficulties in, at least for regulators, to actually interpret and accept the data. Then let's talk about the standard and non-standard situations. And this is, I know this sounds like beating around the bush and trying to squirm around things, but when you develop orphan medicinal products and medicines for children, those are supposed to be taken as standard situations because you would require the best possible level of evidence. But it doesn't mean that you will have to have the Rolls-Royce development of 2 large clinical phase 3 trials. So development of medicines in these situations, in orphan conditions and for children, follow the principles of evidence-based medicine, but we have to take account of the limitations, and therefore we often require non-standard solutions. If your standard solution is to do the parallel groups randomized control trial in an orphan condition, that may simply out of reach. So you have to have an alternative or alternatives. These are just the levels of evidence you all know already, but the questions you would want to ask. Is it possible to do a randomized control study? If it is possible, is a small randomized control study better than a larger study without a control group? And in most of the cases, scientists, investigators, regulators will tell, yes, it's a better alternative to do a small randomized control study than to do an open label study without control arm. Because you will have interpretable, you will have the yardstick in that study. It is seldom that we see treatment effects that knock your socks off. And it is easily risking the whole acceptability of the development of the new treatment if you don't aim for the highest ladder of evidence. Adaptive studies then, a buzzword, adaptive studies. Everybody is talking about adaptive trials. Now what do they mean? It means that the design of an ongoing study can be adapted or changed based on predefined rules. And the emphasis really is on predefined. In early development phases, when you're doing your phase 1, your phase 2, you're exploring, you're wanting to know if there's a hint, if there's a promise of treatment effect, you can do adaptive trials. They're actually encouraged by the regulatory authorities because there are an effective, efficient means of finding out whether there's a promise that could be then tested in a confirmatory study. And you can do your dose ranging as well with adaptive designs. But important is that you have to pre-plan what adaptations are possible. And the adaptations are done based on interim analysis. And the adaptations can introduce changes in the number of patients enrolled in the study. You can add or you can discontinue treatment arms based on what you observe. If one of the treatment arms is showing absolutely no promise of any effect, you can discontinue that. Or if one of the treatment arms is showing an effect but is really showing bad safety and tolerability, you may want to discontinue that as well if the other treatment arms are showing some effect and an acceptable level of safety. You can also do what we call seamless adaptive studies. Well, seamless means that in the typical case, the conventional case, you do your phase 2 study. You have a period of not doing any trials. You have a white space between phase 2 and phase 3. you analyze your phase 2 data before you begin your phase 3. But in a seamless trial, you begin with phase 2 and you move on without any pause to phase 3. So you have an interim analysis between at the end of phase 2. You look at whether you've found the right dose, whether there's acceptable safety and so forth, and then you continue directly to phase 3. And in the best case, you can get much more data from a smaller number of study subjects by doing this sort of a trial. But it's logistically really demanding, and it has to be extremely well planned. But certainly something to keep in mind every time when you are discussing ways or strategies to develop new treatments for rare conditions, adaptive designs and potentially seamless adaptive studies. You can always ask the investigators, have you thought about that, and what are the benefits, and what are the pros and what are the cons, and then tell your own opinion. Sequential studies. These studies enroll patients and they analyze as they go. They analyze continuously the data that emerge from the study, and they do that until a reliable positive or negative conclusion about treatment effect is possible. That's often that is also called the Bayesian approach to analyzing studies, and it has its advantages, particularly in the early development and in situations where you can observe the treatment effect quickly. And there's actually been one not very recent, but one centralized procedure approval of a drug which was originally studied in this way, and that is ibuprofen for a rare condition called patent ductus arteriosus, so inborn cardiovascular condition. And it was studied in this way. In other words, they had small cohorts of patients, they gave the drug at different doses, and they analyzed the data as they emerged. It was completely open label, and they analyzed the data and continued the study, and they would until they were sure that the dose they had that they had identified the correct, effective, and safe dose. And then they took that dose to further studies to confirm the result. And this is what you can do also in situations where the treatment effect emerges relatively quickly, like in a matter of a couple of weeks. We could envisage that, for example, gene therapy for hemophilia, you could give the gene therapy to patients at different doses. You would probably be able to observe the treatment effect around 2 to 4 weeks. And you would evaluate the data as they come and then decide if it's necessary to go up or go down with the dose. And you would receive also initial proof of concept that the gene therapy is actually working as it ought to be working. So a very effective way of approaching rare conditions. The key factors in studies in small populations is that the data collection has to be really standardized, prospectively planned in the protocol and adhered to. You will always have the problem of study dropouts, and it's important to try to minimize the risk of study dropouts as far as possible. And there you will easily have to strike difficult balances, because if you have a study in a small population, you will want to collect as much information as possible. But then you will have to remember the burden of that study participation. If you have to fill in forms corresponding to 10 or 12 different instruments each day, that really becomes a burden. Or if you have to undergo several invasive procedures. Understanding the natural course of the disease and how this may change over time. I mean, this sounds so common sense, but it's so commonly forgotten and not really sufficiently taken into account in designing trials and also in interpretation of the trial results. So natural history studies are really important for each and every rare condition to take into account when you want to interpret the results. It's important to understand the sources of variable response to therapy. And one example that I would like to quote is in the field of enzyme replacement therapy. If the enzyme replacement therapy is shown to decrease heart complications in a study that doesn't have a control group, and we want to use historical comparisons, we end up having maybe questions about, is the historical comparator group really comparable to the study cohort? Because the baseline, the supportive therapy that these patients are receiving may have changed over time and may have actually impacted more the risk of developing certain heart complications. So it's difficult to disentangle the actual contribution of the enzyme replacement therapy in this context without a control arm. These are just different ways to measure efficacy, and basically we already have been through all of these different options. In rare conditions, you will oftentimes have to rely even on biomarker data. But if the results are really positive, and if you have taken the totality of evidence together, strong evidence of there being clinically meaningful treatment effects, you may get a marketing authorization, and then the expectation is that you provide further information based on hard clinical outcomes later on after the approval. We're increasingly seeing drugs approved based on biomarker data, strong biomarker data, followed in the post-authorization stage by a phase 3 trial showing an effect on phenomena such as kidney transplant, hemodialysis treatment, and so forth. So just some of the questions that you have to answer, some of the questions you have to ask and find a reasonable answer to when you plan trials in small populations. What is the appropriate and ethically possible and medically feasible comparator? Can you use a historical control group? Is it possible to do a study without control arm at all? And as I mentioned, cancer is typically a situation in advanced cancer when you don't have any established therapies. Is an area where you can do studies without control group. Just to conclude, we always prefer the randomized, controlled, blinded studies, and I think this is going to be the situation for the decades to come. There are increasing ethical issues and debates on use of placebo. That's recognized by all authorities. But it's important when you strike the balance to realize that there are ethical issues as well if you're using a study design that won't give you the answers that you need. Sometimes it can be more ethical to use placebo than to use a comparator in a non-inferiority setting when the outcome cannot be really scientifically interpreted. The use of active comparators will increase, and we're going to be hearing about health technology assessment in a short while, after a short while, and this is probably going to be tackled in that context as well. It's really important for us to understand where the new therapy fits in, how it performs in comparison to what we have. We often hear that the study, reaching the highest ladder of evidence possible is really, really cumbersome. And what the regulators propose may not be practical. It may turn out to be impossible. What the regulators propose to you may seem really like the Rolls-Royce development, but it may turn out to be possible to perform at the end of the day. or the other way around. So you need repeated dialogue between the sponsor, the investigators, the patient groups, patient advocates, and the regulators, and that is why it is important to understand the terminology used, it's important to understand the regulatory procedures, and it's important to understand the basics of clinical development strategy. I believe this was the last one, so sorry for not quite making it almost 10 minutes over the time. If there are any immediate questions, yes please. Sorry, I'm not sure if I understood. Are there any standardized criteria to assess? Yes. Well, there are all sorts of important documents that everyone should pay attention to that relate to how you should be designing clinical trials, how you should evaluate the results. And I would like to refer you to the European Medicines Agency website when you have the clinical guidelines corresponding to specific clinical situations like diabetes, hypertension, studies in small populations, pediatric studies. Then you will have a great number of international conference on harmonization guidelines that deal with the general design principle, how statistical analyses are done, how to choose the control group, and so forth. So there's really, quite a large volume of literature in the form of guidelines available there. That's our supposed to be followed by the pharmaceutical industry, by investigators who want to have their products eventually licensed, or small companies wanting to have their products eventually licensed, and they make excellent reading for everybody. And I think the glossary that you have in your blue files will help you through many of the aspects of regulated jargon that you will come across. Okay, thank you very much.